How do breast cancer biopsies uncover molecular tumor secrets? Imagine trying to understand a complex puzzle by examining just one piece. That's what a biopsy does for breast cancer. It takes a small tissue sample to reveal the tumor's hidden molecular story. When doctors suspect breast cancer, they often perform procedures like core needle biopsies, which use a hollow needle to extract tissue from the suspicious area. This method is popular because it provides enough tissue for detailed testing while being minimally invasive. Sometimes they might use fine needle aspiration, which collects cells, or even remove the entire lump through an excisional biopsy if needed. The goal is to get a representative sample of the tumor to analyze its molecular makeup. Once the tissue is collected, advanced laboratory techniques come into play. One key method is immunohistochemistry, which uses special antibodies to detect specific proteins on the tumor cells. These proteins include estrogen receptors, progesterone receptors, and HER2. Knowing whether these proteins are present helps classify the cancer into subtypes, like hormone receptor positive or HER2 positive. This classification guides treatment options, because tumors with certain proteins may respond better to targeted therapies. Another technique is in situ hybridization, including fluorescence in situ hybridization, which looks directly at the genes inside the tumor cells. It helps confirm if there are gene amplifications or translocations, especially when protein tests are borderline. For example, if a tumor shows a borderline HER2 protein level, ISH can verify if the HER2 gene is amplified, ensuring accurate diagnosis. Next-generation sequencing is a powerful tool that reads the DNA of the tumor. It identifies mutations, insertions, or deletions in hundreds of cancer-related genes. This information can reveal if the tumor has mutations that make it resistant or sensitive to certain treatments. For instance, mutations in the ESR1 gene can affect hormone therapy effectiveness, while alterations in the PI3 kinase pathway might suggest different targeted options. Proteogenomics is a newer approach that combines protein analysis with genetic data. It helps researchers understand how genetic mutations influence protein behavior in the tumor. This combined view can explain why some tumors respond differently to treatments, even if they look similar under the microscope. It also sheds light on tumor heterogeneity, which is the presence of different cell types within the same tumor, influencing how the cancer progresses and responds to therapy. All these molecular tests are crucial because breast cancer is not one disease but many subtypes with unique genetic and protein profiles. By analyzing the biopsy tissue, doctors can determine the tumor's molecular subtype, identify specific genetic mutations, and predict how it might respond to different treatments. This personalized approach helps tailor therapies to each patient, increasing the chances of success and reducing unnecessary side effects. It's important to remember that a biopsy samples only a small part of the tumor, so it might not capture all the diversity within the cancer. Despite this, molecular testing of biopsy tissue remains the foundation of modern breast cancer diagnosis and treatment planning. It unlocks the tumor's secrets, guiding doctors toward the most effective, individualized therapies. Always consult healthcare professionals for personalized advice, as each tumor and patient is unique. In the end, these molecular insights are helping us move closer to more precise and effective breast cancer care.